In our previous lesson, we saw that the DNA double helix adopts a particular conformation in its secondary structure. Let's look more particularly at the details of this arrangement in order to understand the stabilizing factors. The most common physiological form of DNA is referred to as b-DNA, pictured as a ball and stick model on the left and a space-filling model on the right. The helix is right-handed with an inner diameter of 11 angstroms, and this results in 10 base pairs per turn of the helix. Another observed form of DNA is called a-DNA. It is still a right-handed helix, but as you can see from the figure below, it is thicker than b-DNA, having 11 base pairs per turn of the helix. This has only been observed in the lab and would not necessarily represent a native conformation. Another naturally occurring form of DNA is z-DNA. It is a left-handed helix and typically results from alternating purine and pyrimidine bases. It may play a role in the regulation of gene expression. Looking more closely at the secondary structure of z-DNA, we see that it results from flipping one side of the backbone by 180 degrees without breaking covalent bonds, and that it still maintains the hydrogen bonding contacts. Thus far, we've only considered the hydrogen bonding interactions of DNA. However, there is yet another and more important means of stabilizing the double helix, and that is through the interactions of the bases that are internal to the helix. Recall that these bases are very hydrophobic and would therefore interact via van der Waals forces. Also remember that these bases are conjugated and therefore have pi orbitals associated with their aromatic rings. As the bases stack in the double helix, pi orbitals overlap, resulting in greater freedom of movement for electrons within the pi cloud, as shown here. This is a primary stabilizing factor. In standard b-DNA conformations, each base is rotated by 32 degrees with respect to each other. This provides the best arrangement for base pairing interactions, but is not optimal for base stacking. The twist angle is shown in the figure below, as well as the fact that the bases within the minor groove are in close contact with water, which is at variance with the hydrophobic effect. For these reasons, many bases within DNA adopt what might be described as a propeller twist, shown below. This results in base pairing that is less than optimal, but improves the base stacking interactions. This also serves to eliminate water from contact with the bases in the minor groove. Thus we see the hydrophobic effect dominates the formation of the standard b-DNA double helix.